Hello my dear students, welcome to the MS Science Academy. Myself Meenu Srivastava, and today we are going to discuss about the oxidative phosphorylation. Oxidative phosphorylation, and before starting the oxidative phosphorylation, first we will discuss about the substrate level phosphorylation. In my future lecture we will discuss about the substrate level phosphorylation in detail, but today we have to compare about the substrate level phosphorylation and the oxidative phosphorylation. In glycolysis, in the Krebs cycle, in many other reactions, we have seen that in the different reaction, if the ATP is generated directly, so this ATP and the directly generation of ATP comes under the substrate level phosphorylation. But if the energy is generated in the form of NADH and the FADH2, for the cell it is very hard to utilize. So for the cell utilization purpose, we have to convert NADH and FADH2 into the form of ATP. So the conversion, for the conversion, we have to take the NADH and FADH2 into the electron transport chain, which is present in the mitochondria. Okay, so in the mitochondria, there are the 4 complexes, there are the 5 complexes. In the ETC, we have studied already. Okay, so in the 4 complexes, the protons are generated and electronic shuffling will be there. So, due to the protons generation and the electron shuffling, the NADH and the FADH2 will convert into the ATP by the help of complex 5. So, before starting the oxidative phosphorylation, we have to see the structure of the complex 5. Okay, so the complex 5 has the 2 subunits. One is the F0 subunit and second, that is the F1 subunit. Okay, F0 subunit and the F1 subunit. The F1 and the F0 subunit. The F0 subunit is rotating subunit, we will see. Okay, it will rotate. Okay, and it is embedded in the inner membrane, inner mitochondrial membrane, yeah, or you will say this inner mitochondrial membrane, IMM, inner mitochondrial membrane. So the membrane nature is the lipophilic. So the F0 unit nature is also the lipophilic or hydrophobic. Okay, it lies in the inner mitochondrial membrane. It consists of 10 to 14 channel proteins or C proteins. Okay, so such kind of the C proteins I have drawn here. So these channel proteins, okay, these channel proteins are present. These are the polypeptides, okay, and they act as a proton channel. They act as the proton channel. In the F0 region, they are the sum, and they are the one A subunit also, which help to connect the F0 region and the F1 region. Here the C subunits and it connects with the F1 region, but here the connection, suppose that it's a stalk, this is the C units and it's a A subunit. Okay, so C subunits, C proteins, channel proteins and A subunit combined form the F0 unit. Okay, now the F1 region is present in the matrix part. Okay, and it is a catalytic unit because only in this unit the ATP will be synthesized. Okay, how it will be possible we will see, just later on we will see, so just stay with us. The F1 region is present in the matrix site, so its nature is the hydrophilic. Okay, it lies towards the matrix and it consists with 3 alpha and 3 beta, that is the hexamer ring. Alpha 3 and beta 3 also we will say this, 3 alpha and 3 beta unit, alpha 3 beta unit, alpha 3 and beta 3 here is present as a hexamer ring. Here this is a stop, this is a C unit, this is the A, here the alpha beta. Okay, so here the alpha and beta are present in the alternative form. Okay, so alpha 3 and beta 3 present in the form of the hexamer ring. Okay, so here alpha and beta are present, but beta is mainly responsible for the synthesizing the ATP. Alpha is not responsible, but only beta is mainly responsible for the synthesis of ATP. Okay, so F1 region is made up of 9 subunits, 3 alpha, 1, 2, 3, 3 beta, 1, 2, 3, 3 alpha, 3 beta, 1 gamma. This is a stalk. This is the stalk which is present between the, which is the part of the F1 region. Okay, so here, which is the present in between the F1 unit and the F0 unit. Okay, so here the alpha 3, beta 3, 1 gamma, 1 delta, 1 gamma present in the stalk, 1 delta present in the association with the hexamer ring delta, and 1 epsilon. Epsilon is associated with the stalk, 1 epsilon. Okay, so 3 alpha, 3 beta, 1 gamma, 1 delta, and 1 epsilon. Okay, gamma and epsilon, they form the stalk. Okay, and which runs through the inner cavity of hexamer ring. Okay, so these are the hexamer ring. Suppose that these are the hexamer ring. So this stalk, which is composed with the gamma polypeptide chain and the epsilon polypeptide chain, this ring, this stalk will just go through, runs through the inner cavity of the hexamer ring, which is composed with the alpha 3 and beta 3 subunit. Okay, now the delta subunit is hold the alpha 3 beta 3 hexamer ring. The delta subunit hold the alpha 3 beta 3 subunit. Okay, now the region, which region is rotating region and which is the stationary region? We will see in the channel binding mechanism, by the rotation there is the one proton motive force is generated and will be generated, and the ATP synthesis takes place. But how is it possible? We will see later on. But what is the rotating region consist of? The rotating region consists of these channel proteins and gamma and epsilon stalk. Okay, so the C proteins, the stalk is composed with gamma and the epsilon. Okay, and the stationary region or remaining stationary region, all the other regions, all the other subunits, all the other polypeptide chains. Okay, like A, 2B, 2B polypeptide chain, 2B polypeptide chains, and alpha 3 and beta 3. Okay, these are the other subunits which are remaining stationary. So the rotating region is behind that S, C rings and the gamma epsilon stalk, and the stationary region is the other units. Okay, so here is the F0 region, C unit, around 10 to 14 C proteins, channel proteins, C units are present, and this is associated with the A subunit, A polypeptide chain, and this A polypeptide chain is combined with the F1 region with the help of 2B polypeptide chains. Means here this is A and 2B polypeptide chains are present, and these 2B polypeptide chains are present, means 2B polypeptide chains. So don't get confused. Okay, polypeptide chains. Okay, and now this is the delta. The 2B polypeptide chains is associated with the delta, and delta is associated with the hexamer ring. Okay, so F0 region, C units is associated with the A is associated with the 2B subunits, yeah, B polypeptide chain or B polypeptide chain, then delta, then hexamer ring, and hexamer ring associated with the stalk. Stalk is consisted with the gamma and epsilon, like this. Now we will discuss about the oxidative phosphorylation, how it will take place. Let's see. Now come to the oxidative phosphorylation complete definition and its mechanism part. Here now come to the definition part. The transport of electrons through the ETC. As we have seen in the ETC, the electrons are shuffling is linked with the release of free energy. Okay, so by shuffling of electron, the free energy is generated in the form of ATP by coupling with ADP and the inorganic phosphate, and this term is known as the oxidative phosphorylation, that is the OP. OP represent, I am here just like oxidative phosphorylation. In the short form, I have described that in the complex 5, the ADP and the inorganic phosphate is formed as the ATP. There are so many mechanisms which are proposed for the oxidative phosphorylation, but the most acceptable theory, that is the chemiosmotic theory, which is proposed by the Peter Michel in 1961. Okay, so this is the chemiosmotic hypothesis, the most acceptable theory, which can explain up to the very much extent the generation of ATP. Here, as we have seen, as I have already told, that the inner mitochondrial membrane is impermeable to the proton, and in the electron shuffling, in the electron transport chain, the protons are sent toward outer side of the inner mitochondrial membrane. So there is the accumulation of more protons or more hydrogen ions on the outer side of the inner mitochondrial membrane. So the matrix is deficient with the proton. So the outer side of the inner membrane is very much rich in the proton, but the matrix site is deficient. So there is a one proton gradient, or the electrochemical gradient is created. Okay, now we have to manage, we have to balance this gradient. Gradient means just difference. So don't get confused about the gradient. Some students are getting confused, what is the gradient, what is the gradient? This is the main confusing part. So do not confuse, the gradient means the difference, means the proton difference is created. But in the biological term, you have to use the gradient. Okay, so the proton difference is created. So here again the protons have to shuffle back into the matrix. So the protons just sent back through the into the matrix via the complex 5. But this is the most important question. Okay, we agreed about that the complex 5 is responsible to send the protons from outer side of the inner membrane to the matrix side. But how? So we are not going into the deep, but there are some half units, hydrophilic half channels present on the A subunits, and the C subunits having the aspartate 61 units, aspartate 61 residue. Aspartate 61 residue is present. Okay, half channel means half channel here. Suppose that this is the A subunit, it is having the half channels. Half channel means the channel is not completely embedded. Okay, it's some part is present on the upper side and some part present on the downside of the membrane. This is the half channel, and this is present on A side, A subunit, and A subunit, where the A subunit, this is the stalk, this is the C subunit, and on the C subunit, A subunit is present. So A subunit is containing the half channels. Okay, so these half channels are mainly responsible for the shuffling, and the C subunit is having the aspartate 61 residues. These are also responsible for the shuffling of proton, and this shuffling leads the conformational changes in the beta rings, beta polypeptide chains. There's a 3 beta polypeptide chains, and these beta polypeptide chains or beta ring are having due to this proton motive force, they are just generate the conformational changes, and this lead the we can say this lead the generation of ATP. Okay, here in response to the proton flux, the gamma subunit rotates. Here when just this proton shuffling via this half channels and the aspartate residues, when they are just going through the stalk is composed with the gamma subunits, so the rotation of stalk will take place. So the rotation of the gamma unit when the stock is when the gamma subunit is present in the stock, the gamma subunit takes place the rotation. So here when the rotation takes place, the beta subunit having the conformational changes. Generally the beta subunit has a distinct states. One is the tense or the tight, second is the open, and third is the loose. In the tight or the tense state, the tight state is just I will start the loose. In the loose state of the beta, in the loose state, the ATP and the inorganic phosphate become trapped. And the tight or tense state, this inorganic phosphate and the ATP brought close to form ATP. And in the open state, the ATP is formed, is released, and new ADP and inorganic phosphate set can bind. So here I have drawn some diagram that this blue I have made the alpha subunits. Alpha subunits is having the ATP, but they are not participating in the ATP formation. But the catalytic unit is the mainly beta. So beta is mainly participating in the ATP formation. So here I have discussed I have made in the with the red color that the open, O for open, L for loose, and T for tight or tense. In the loose state I have discussed that ATP and inorganic phosphate trapped. Now just trapping the ADP and inorganic phosphate, and in the tight state the ATP formation takes place when the ADP and inorganic phosphate come closer. And in the open state, when the ATP is released, the new ADP and inorganic phosphate come to take the chance. Now, what will happen when the stock will move? The 120-degree rotation counterclockwise. What is the counterclockwise? Anti-clockwise. So here, when the tight beta ring is just 120-degree rotation is reached towards the loose state. Here tight reach the loose state. This is the tight, and loose state reach towards the open state, loose state, and open state reach towards the tight state. Okay, so this kind of the shuffling, this kind of the shuffling will takes place anti-clockwise. Okay, so here in the tight state, when it just reach towards the loose state, the tight state is having the ATP. Okay, it come close the ADP and the inorganic phosphate, and just make the conditions to form the ATP. In the loose state, the ADP and inorganic phosphate just trapped, and in the open state, these trapped ATP formed just here. In the open state, the ATP formation takes place, and as the ATP formation takes place, the ATP is released. So here the open state remain empty. Okay, so here the open state remain empty. Now again the new ADP and the inorganic phosphate set come and takes place the open state. Like this, the conformational changes, the ATP is released. Now we will discuss that in how much NADH and in the NADH how much energy is generated, and the FADH2 how much energy is generated. So as we have seen, this cycle leads the conformational changes of the beta subunit. Okay, so by this the conformational changes, the ATP is generated. Okay, so by the generation of ATP, okay, so due to the conformational changes of the beta subunit, ATP is generated. And now we will do the calculation part, that NADH and for the NADH and FADH2. As in the NADH complex 1, 4 protons are involved, complex 3, 4 protons are involved, and the complex 4, 2 protons are involved. So the total 10 protons. And you have to keep in your mind that for the synthesis of 1 ATP, 4 protons are required. So here are 10 protons. So 10 by 4, 2.5 ATP. So for NADH, 2.5 ATP is generated. But previously the concept was there that from 1 NADH, 3 ATP will be generated. But now new concept is there, 2.5 ATP. Now come to the FADH2. As we have seen in the FADH2 complex 3rd and the complex 4th are involved, 4 protons and the 2 protons. So here the 6 proton by 4, it's a 1.5 ATP. So like this in the NADH, 2.5 ATP, and in the FADH2, 1.5 ATP will be generated. So this complete mechanism is known as the binding change mechanism, and the complex 5 is known as the smallest molecular motor. Why is it so? As you have seen that generation, synthesis, rotation, there are so many things will take place on the in the complex 5. So that's why it's the smallest molecular motor. I hope you enjoyed my lecture. Any doubt, kindly ask in the comment section. Otherwise, please like, share and subscribe. Thank you.